So you can see my screen? Everything's good?
Looks good.
Okay. And yeah, if anyone has any questions, just feel free to ask, and I'll send my email in a bit. And you could ask further questions. Maria and Krista, feel free to jump in at any time and specify stuff for the directory if you would like. And so I'm guessing most of you guys are in undergrad. All right, so I guess we'll talk about viruses today. My name is Pradeep Ambrose. I'm along with Maria and Krista. I was co-mentored during my PhD by Chris Mason and Charlie Rice. And bitter news, I don't know if you guys saw it, but Charlie won the Nobel Prize on Monday. And he, along with Harvey Alter and Michael Houghton, won it for hepatitis C. Harvey and Michael in the 1980s, I think 1989, they discovered hepatitis C and developed a test to figure out what if the blood supply has it, because they noticed that people getting blood transfusions all of a sudden started getting this weird hepatitis that they initially called non-A, non-B, because they only knew hepatitis A and hepatitis B at the time. And it took a while to figure out what it was. And Harvey and Michael figured out what it was and developed a test so that the blood transfusions can be tested for it. Sort of getting rid of that as the major source of hepatitis C transmission. And then Charlie, a few years later, in the mid 1990s to early 2000s, so about a decade before I joined the lab, but I'll take partial credit anyway for the Nobel. No, just kidding. His lab figured out, at the time, treatment of hepatitis C was really difficult because you had to do it in chimps. Chimps were the only organism that could, only animal that could replicate hepatitis C. No one still knows where it came from or how it appeared, because now it primarily transmits through blood, but at some point it had to come from a different animal or something, and no one still has any idea. It grows in chimps a little bit, but and Charlie and a few others, one figured out how to get it to grow in cell culture, which is, I guess, here. If you guys could see my mouse pointer. And that just allowed you to work on it in a lab instead of in, a zoo, if you will. And that just accelerated the pace of discovery. He made a bunch of other discoveries on what hepatitis C does, but the cell culture system that he developed was used as a screening platform to discover sofosbuvir, which is a drug that came out a few years ago, I think 6 years ago or so, that cures it 99.9% of the time. That's Gilead's big drug. And I was in the lab about a year into them, about a year before that drug was released, and it was a pretty cool process. And I had no idea when I joined the lab. He's just just a really nice guy, and I like the environment, and I had no idea he had done this, because okay, so I guess I'm gonna talk about viruses in the context of evolution a little bit, because that's what my research is in, and I think it puts it into a good context relative to other organisms, and it'll give you a bit of an idea of what research is going on. As Darwin said, natural selection is nature-born variation times the geometric rate of increase times the struggle for existence. What does that mean? Nature-born variation, in his term, he didn't know it at the time, but now we know it's differences in DNA, differences in your genome that make you slightly different. And the geometrical rate of increase is every generation has more, every generation gives birth to more organisms than there are in the previous generation. So if one tree produced one tree ad infinitum, then it wouldn't work. You need and that tree never died. It's every living thing produces way more offspring than there is space to handle. Doesn't really apply to humans anymore, but in general, in nature, that's the case. Times a struggle for existence, which is where a large number of those offspring die, and the natural born variation that makes some more fit and others less fit just changes the gene pool as things move on. And the question is, how does that apply to viruses? So I guess the agenda today is I hope to show you that viruses provide an excellent tractable system to view evolution. That was my research, but I think just like a good context to put basic virus stuff into. Virus genetics, in addition to like just being of interest to biologists, has significant impact on human and animal lives and agriculture. I don't need to really I pulled this from an old slide where, at the time, no one really cared. So you had to be like, hey, viruses are important. Now it's not, I think everyone kind of gets it. And the current genomic era with next generation sequencing and bioinformatics, I'll explain that a bit later if you don't know what that is, allows for robust virus genetics at previously unseen scales and resolutions. So one aspect of that is that it takes significantly shorter to discover what a virus is when an outbreak happens. It used to take months, and now it could be done in a few days. And 10 years ago, it took a few weeks. 20 years ago, it took about a month or two. And that's true of, that's true comparing SARS-1 to SARS-2 or SARS-1 MERS to SARS-2. If you even look at the same family, it has accelerated tremendously. So the key concept here is fitness. What is fitness? Biology has a really simple assay for any given genome. You could ask, does it live and does it reproduce? So it's pretty simple. For humans, it's like, does it live? Yes, no. A genetic defect such as trisomy 16 is the biggest cause of miscarriages, and that's just not alive. But the more important thing for biological fitness is, does it reproduce? So for example, yes, your parents. No, there's something called the Klinefelter syndrome where people are perfectly fine. And they're alive and they like barely, a lot of times they don't even know they have it, but they cannot reproduce. They're infertile. So biology of fitness means basically how, if you're reproducing in this context. And viruses, does it live as metaphysical? Whatever. You could think of it whichever way you want. And does it reproduce? We really think of it as, does it replicate? So one virus goes into a cell, and in general, that cell can produce 10,000 new viruses. So it has a high dynamic range of fitness. You just divide like 10,000 by 1. If 10,000 come out and they're all alive, then the fitness rate is 10,000. If 10,000 come out and 5,000 are alive, the fitness rate is 5,000. So there's a gigantic dynamic range to quantify and do research on there. And the key there is that lowering viral fitness, lowering this reproduction rate, is the key to all vaccines and treatments. So I guess what are viruses? It's a genome and a protein code, basically. And there's 4 genotypic classes. Viruses are weird. All if there's one takeaway from this entire lecture that I think you should have is that it's that viruses are weird. There's 4 genotypic classes. All life outside of viruses are all double-stranded DNA. So you don't even think about this in any other context. But viruses can exist as double-stranded DNA, single-stranded DNA, double-stranded RNA, and single-stranded RNA. And within single-stranded RNA, 2 different types. And then it can get even weirder. But for now, we're going to focus on this. Like, it can be partially double-stranded, like half double-stranded RNA, half double-stranded, half RNA, half DNA. But like that's getting to like some really weird ones. If you guys want to know a really weird one, look up hepatitis B after. That's the weirdest virus, I think, in general, that we work on in the lab. So there's 4 different genome types, and of RNA could be 2 different types. And I'll explain what that means in a second. But here, in general, you have, when it's outside of a cell, you have the genetic material in the middle somewhere. It's surrounded by a protein coat always. The protein coat, and sometimes enveloped viruses. Yeah, there's another distinction. You have viruses that are enveloped and viruses that are non-enveloped. And the big difference there is that if viruses bud from the cell membrane, then it's covered by a piece of the cell membrane. It usually like pushes its spike proteins. So all viruses have proteins that jut out at the end for it to stick to its next target cell. And if a virus like buds off from the cell membrane, then it will generally have an envelope, which is just the lipids that are part of like your cell membrane that it just like popped out from. And there, SARS-2, COVID-19 virus is an example of that. The key there is that the cell doesn't need to die for the virus to get out. It just, like, as soon as it replicates, it just buds off as the cells are still alive, and the cells could slowly die, the cells could never die, whatever. On the other hand, non-enveloped viruses are just a protein shell, and polio is an example of that. And the way it gets out of the cell is by like replicating, and the cell just explodes, and then it gets out. So it doesn't have an envelope because it doesn't go through the plasma membrane of the cell. And that also is how you extract. Like, if you're making envelope viruses, really easy. You just like grow it on cells and then pour the fluid that's on top of the cells, and there's your envelope virus. If you're in the lab, if you're making non-envelope viruses, because they're still trapped in the cells, you gotta take all your cells and then freeze thaw them really quickly in liquid nitrogen a few times to like force whichever cells that haven't exploded yet to explode to get your virus. And that's a large generalization God damn it. That's a large generalization for a variety of different envelope sizes, shapes, etc. And now we're going to go a little bit into the different genome types of viruses, where viruses are really, unless we find, we haven't found organisms outside of viruses that have genomes made of these different types. So like when you think of viruses, first you want to think of genome type, because generally in all of the forms of life that we know so far, it's double-stranded DNA, but in viruses, it could be any of these. There you have single-stranded DNA and double-stranded DNA viruses. You have parvovirus here. This infects dogs. and a lot of animals causes distemper virus. Double-stranded DNA, you got pox virus, smallpox, adenovirus cause a lot of colds. I'll talk about this later, but some of the COVID vaccines are put into an adenovirus backbone. Herpes, HPV, papillomavirus that I think you guys are now getting vaccinated for. And then their DNA just is copied to DNA to make the new virus particles, to make its offspring. Then you have, oh yeah, this is a weird one. You have double-stranded RNA that goes through an RNA phase. That's hepatitis B. Hepatitis DNA is hepatitis B. And it's just really weird. Oh, so on the left are viruses that infect vertebrates, including us. On the right are like all the other life forms that are infected by this family of virus that we know of so far. There's an order of magnitude more that we don't know. And then plus-stranded RNA that's reverse transcribed, that's HIV. Retroviruses, including HIV. So the virus in the shell. So when we talk about like the genome here, we're talking about what's in the shell. Once it goes in, and then when we talk about like how it reproduces, we're talking about how more of that genome is made for the next batch of shells once it's inside of a cell. Does that make sense so far? And then you have RNA to RNA, where, RNA goes in and it just directly copies itself to RNA. There's no DNA step there. And you have the plus-strand RNA, which is corona viruses. You have all the mosquito-borne viruses here for the most part. You have polio and the picorna viruses. You have arteriviruses that are sort of close to coronaviruses, cause a lot of animal diseases. Picorna is funny because when Charlie had his virology course, he invites a professor to give a talk about, each, like whoever is the leader in the field to come give a talk about their virus family. And this one guy, he discovered a lot of he was a structural biologist, so he discovered what a lot of these protein codes look like. He's been at Harvard for like 50 or 60 years, and he always goes on a rant about how much he hates the name picorna because it's pico-RNA, and it stands for small RNA. And he just thinks it's the stupidest name on the planet to call something small RNA virus, and then just fuse pico-RNA to picorna. Anyway, I don't know why I went into that. You got double-stranded RNA, which is reovirus. And then you have negative-stranded RNA, which includes Ebola and, Ebola. One similar to Ebola are filoviruses. You have rhabdoviruses, which is rabies, things of that nature. And about 70% of common colds are also in the pico-RNA family. Then you have, this is just to show you electron micrographs of what these look like. These ones as tails are viruses that infect bacteriophages. No vertebrate virus looks like that. Then you have tobacco mosaic virus. You have, this should be a rhabdo. Yeah, rhabdovirus, VSV, that's very closely related to rabies. And you have rotavirus that causes diarrhea. So now we're going to go to the Baltimore Classification system. This is generally the classification system used for, that single-stranded RNA, single-stranded DNA, double-stranded RNA, double-stranded DNA. was discovered by this guy, David Baltimore. And I just have a picture of David here because he was on a virus Zoom chat recently, and we just thought his Zoom background was pretty funny. But so yeah, he's at Caltech now. I think semi-retired, but he came up with this, and it's been used since. And he won a Nobel Prize for this also. But generally, to make viral proteins, including the shell and stuff that copies itself, you need to get to positive sense mRNA. And positive just means that is the strand that protein is made from. Negative just means it's complementary to the strand. Like when it's in double-strand form, negative is the other side. So protein cannot be made from the other side. It's just the reverse complement to the side protein is made from, where the triplet code makes sense for its protein product. So then it's just what's in the shell and how it gets to positive strand mRNA inside the cell to make its protein. So like what's in the shell? You have positive strand DNA, which is one form. And that can, once it's in the cell, it makes double-stranded DNA. That double-stranded DNA is made into mRNA by cellular machinery, and voila. You also have double-stranded DNA that just directly gets made into positive-stranded mRNA inside. You have double-stranded RNA, which just like splits once it goes in. And proteins made from the positive strand mRNA. You have minus strand RNA that is made into double-stranded, that is just polymerized into double-stranded RNA, and then the other side they split off, and then the other side is the plus-strand mRNA that makes protein. Yeah, positive strand RNA that goes in, makes double-stranded RNA. The first mRNA makes protein straight up. And then to make more of the positive strands, then it's made into minus strand, and that minus strand is a template for more positive strands to like accelerate protein production and also to make the genomes that go into the shell. And then you have the retroviruses like HIV, where the RNA goes in, it's made not... So when it's made into double strands, it's not copied into RNA like in the positive strand RNA viruses. The other side is polymerized as DNA. And then that DNA, it is made into double-stranded DNA, and that is then made into mRNA. So positive strand RNA viruses is where, as I mentioned, single-stranded RNA viruses is where RNA cells don't have a enzyme that makes RNA into RNA. So viruses bring that in with them. In the case of negative strand viruses, because the first protein cannot be made from the virus itself, it has to carry that protein in the shell. In the case of positive strand RNA viruses, the first genome comes in, makes the protein, and then kicks off the rest of the process. So they all require something called the RNA-dependent RNA polymerase that just copies RNA to RNA. And I'm going to focus on plus-strand RNA viruses because, one, that's what coronaviruses are, and so that's what most people are interested in at the moment. Two, that's what I worked on and is, consequently clearly the coolest virus family. For plus-strand RNA viruses, the RNA is both the genome and the transcript. What is in the shell is what is made into proteins. And just the RNA itself is infectious. As I mentioned, you don't need an extra protein. You could literally just make the RNA in a machine, put it into cells, and you can make it synthetically. You can order it online, put it into cells, and it'll start making the virus. That's all you need. And we've done that multiple times. And I'm going to start going into disease phenotypes a little bit. So basically, they run the whole gamut. You have commensal, and this was a study maybe about, I don't know, 8 years ago that just like looked at a large percent of human population of children and adults to see like how many commensal viruses they just have in them. And when you're a child, when you're a baby, you basically have very few. And then as a child, just from like playing around and human contact, you start getting a bunch of these. And and these are antibodies. And then as an adult, with more human contact and as you start having sex and stuff, then you just have like a whole bunch of viruses inside you all the time. Um, and what's interesting is like Epstein-Barr, right? Like that's in your nerves, in your spinal cord. And 85%, 90% of adults have that just permanently in your spinal cord. um, Herpes simplex 1, etc. These are cold viruses, so you have antibodies, but they're not like inside you. They don't stay with you. CMV stays in you forever. So like as you get older, you're just like a vehicle for more and more viruses that are just alive and reproducing inside you at some small degree, but you just don't notice because I mean you notice rhinovirus, adenovirus sometimes when you have a cold, but generally like a lot of these you just have the antibodies, you've had them, you don't notice the ones that are in you forever because they don't cause problems like Epstein-Barr and cytomegalovirus. They're just in you forever. They're in your nerves. They don't cause problems unless you have an immunodeficiency, such as HIV. And HIV was discovered because they first noticed that people are dying of things they should not be dying from, that everyone just has. I think Epstein-Barr is called the kissing virus for that reason. Um, and then you have, as you move on, like influenza and rhinoviruses. Sorry, this is supposed to be a chart for both of these. So you have Epstein-Barr cytomegalovirus. Everyone has it. You don't notice it. Then you have like annoying, where you're hot and tired for a week. Most influenza, not all influenza, but most influenza. Then you have rhinoviruses, which is rhino is just a word for nose in Latin. So it's literally rhinoviruses are 70% of cold viruses. They just make your nose run. Sinbis is what I worked on. It's a virus, a mosquito-borne virus that caused a rash. There's my stick figure drawing that I'm very proud of. And then you have deadly, right? You can see my drawing of a cemetery right there. You have Ebola, smallpox, yellow fever, HIV, hepatitis C, and 1918 influenza. Thanks for the thumbs up, Maria. Where it's where it's just like a good percentage. It's different with treatments and vaccines now, but like back in the day, and even so now with like Ebola, there's a good chance you're gonna die. There's no way around it. HIV, that was the case 20 years ago. I mean, if you had it, you died. But now it's very manageable. And like it's kind of like having diabetes. The drugs have gotten so good that you just take the drug every day and you're like basically fine. And then you have, everything in the middle. Zika, dengue, West Nile chikungunya, polio. Polio, interestingly, you guys probably see the images of people in iron lungs and stuff, but polio, that's like a 0.1% of the time that it goes into a lung. Polio is generally a stomach virus. So it's a fecal oral virus. You get it, you have diarrhea for a couple days, and then it goes away. So 99.9% of cases, it is a diarrhea causing virus. In that 0.1% of case, it causes it just goes to your nervous system, and then it causes like all these other complications. But for the most part, it just causes some diarrhea. Hepatitis A, similar thing. The H1N1 flu, it was for the most part, it did kill a small percentage die, not nearly as much as Ebola, where like 40% die. And then our latest virus, SARS-CoV-2, where so many people were infected that a large number of people are dying, but like in terms of percentage, it's not nearly like HIV or hepatitis C before drugs, right? And this, every time I show the slide, I've been doing this for about 7 years now, like more and more things get added, which is pretty interesting. Like Zika wasn't in the first iteration of this slide, and now I had to like go in and add this in yesterday. Maria, do you want to talk about like in the section where it says human disease, like where along this the threshold for that is, or?
Yeah, I think that will be pretty useful for volunteers.
Okay. Do you wanna tell them, because I don't know exactly what the threshold.
So yeah, as Pradeep was explaining, most viruses are within our microbiome, right? So in the microdirectory, we are considering pathogens as those viruses that produce any symptoms like rash, fever, like that usually make people sick. Of course, we know that for any immunosuppressed patient, they're gonna get sick for basically anything. Any commensal will be getting the symptoms. But in this case, for viruses, you'll have to consider if when the host is in contact with the virus, does it get sick? Like that's within the normal process of getting the virus, and then you have to get sick because the virus is like, for example, Ebola. You're very likely to get sick of Ebola, and there is a high percentage of death. So keep in mind that when you are annotating the pathogenicity of the virus. Same for plants. For the plants that get the viruses, and they get like in the leaves, they get spots or the tree dies, or those kind of symptoms are considered as pathogenic in the microdirectory.
I just can I ask? Can you also clarify if the volunteers should be annotating viruses that might affect people who are immunodeficient? Should we be annotating that, or should that not be annotated?
That's a really good question, because we discuss about that a lot, and we decided not to consider any microbe that affects that you find any article saying that this particular microbe, virus, bacteria, fungi affected a immunosuppressed patient. We're not considering that as pathogen, and that's because usually those are within the commensal microbiota. As I said, basically any microbe, even your skin bacteria, are gonna make you sick if you are immunosuppressed. So yeah, thanks.
Okay, so I guess to iterate, the threshold is here. If you guys could see my mouse, if it's commensal and you don't notice, it's not a pathogen. As soon as it starts becoming annoying, it is a pathogen. All right, I'm checking the chat. Does it depend on the cell's machinery to go from RNA to RNA? No, you need your own machinery. I think I went into that. Michael Suarez says #fine art. Thank you, Michael. Best stick figures I've seen. Thank you, Kazi. Rami. Both Michael and Kazi, in the future, if you guys want virology research jobs, I'll remember that. JK. Rami says, can the accumulation of viruses over time potentially cause some of the negative aging effects of aging? We don't really know. Almost everyone just has it, and there hasn't been a huge enough study looking at this as far as I'm aware. So we don't really know. To another point, straight up, your protein coding of the human genome. Going back to, I guess, retroviruses, HIV like makes DNA once it enters, and then it integrates into your genome. And the protein coding percent of your genome is something small, like 5% of your genome actually makes the proteins that make, more or less like stuff that you are. And this process has occurred so many times that 20% of your genome, this is one of the biggest surprises during the Human Genome Project, 20% of your genome are old retroviruses. Like a lot of them are inactive and don't do anything. Some of them are like mildly active and they're controlled by your immune system. People think that immunocompromised people may be negatively affected by some of these like old retroviruses that are just in your genome that are, commensal, so you don't notice, or they've been long dead. But yeah, like you have more viral DNA inside you than you do of proteins and everything else. Finally, you have motor transmission. So, for example, in red are all the mosquito viruses here. It goes mosquito to human. A mosquito bites you. It gets the blood and the virus inside the mosquito's stomach. Then it takes about a few days for that virus to infect so the virus is not just carried by the mosquito. It has to replicate in the mosquito. It has to go through the entire same process as in the mosquito. So it will start replicating, going from the mosquito's stomach to its mouth. And then once it's in the mouth and it's in the saliva of the mosquito, then it'll infect the next human. That's how these work, including Zika, which you guys may have heard about. Let me get rid of the checkbox. And then what is vaccination? Vaccination is just using a low fitness virus to train the immune system so that it acts rapidly when faced with the wild type virus. Wild type virus being a term for just the one in nature. Before genetics, low fitness viruses were generated unconsciously by replicating it outside of humans to accumulate mutations that lower its fitness, and then reintroducing them into humans. And that's the very first instance of a vaccine. Before vaccine, the smallpox vaccine, you had what's called variolation, where people just gave people smallpox itself, and about 5% of them died. That was just, just what you expect. You have a 1 in 20 chance of living. And then if you have it, then you do not get smallpox later. And that was very important for like wars, because they didn't want a smallpox outbreak to happen to soldiers during war, so they just gave them all smallpox before, like in a hospital setting. Thousands of them died, and then the rest of them was like, all right, you're good to go to war. And that was happening for a while. Edward Jenner, who was actually a bird scientist, his like big research project was on cuckoo bird nests. He noticed that like milkmaids who worked on cows like did not get smallpox nearly as much as the general population. And he wondered why. And he found out that they got infected with cowpox, which is, a similar family virus that cows have. And because of that, they didn't really get smallpox. So that was the first vaccine. And vaccine comes from the word vaxa, which literally means cow in Latin. And then the first virus discovered was tobacco mosaic virus. Tobacco was the most important industry at the time. And so they were doing a lot of research there, trying to figure out what's causing our cigarettes to not be made as profitably. And that led to the discovery of tobacco mosaic virus. It was really proven when electron microscopy became a thing. Then you have, similarly, the yellow fever vaccine, which they got from this one guy called the CB. And they passaged it, meaning they just like grew the virus outside the Studa CB. in cell cultures or eggs or, in monkeys, whatever, what have you, whatever they got at the time. And over time, it accumulated 68 mutations compared to the original yellow fever. And that became the vaccine. And that still uses the vaccine. And for that, the guy won the Nobel. This was done in the 1930s. The 1951 Nobel Prize was awarded for making this vaccine, which is just as easy as taking the blood from this guy and growing it in whatever you got your hands on for a little while. Similar thing with the polio vaccine. Both Salk and Sabin vaccines are based on having 3 different types of polioviruses and growing them in African green monkey cells, which is what is used to grow SARS coronavirus 2 now in the lab. They're called Vero cells. And then Salk is inactivated. So like it was a couple passages in monkey cells, and then you add formalin to chew up the proteins a little bit. And Sabin was attenuated, which means you just like kept passaging, which just means kept growing. Passaging is just another word for generations. We just use it in virology. Until it just accumulated a bunch of mutations. So poliovirus 1 there accumulated 57 mutations, poliovirus 2, 2 mutations, poliovirus 3, 10 mutations. They didn't know it at the time, but it was different enough because the same selective pressure is not there in cells that when you reintroduce it into humans, it doesn't cause the full disease, but it does give you the antibodies. And in the genetics era, which I'll cover later, is we kind of have much more insight into this process. I don't know if that makes us better at making vaccines or not, but we have a lot more insight into the process. And I'll show you the variety of techniques that are being used for SARS-CoV-2 now. Just to reiterate, yes, vaccines are a low fitness form now that's all previous vaccines were low fitness forms of the virus. Now you have things like recombinant vaccines where the shell proteins of the virus are put into either a different virus or just introduced by themselves, whatever. Ultimately, they are lower because they cannot replicate. Like the word fitness doesn't really make sense in that context anymore. Yeah, vaccines historically and for vaccines that are the virus itself and not like a part of the virus put into something else, it is a low fitness form. And I guess technically the newer versions, and which I'll get into later, are also lower fitness forms. So what aspects of information allowed it to? So basically, the way they discovered what parts of the virus are important or not is they introduce mutations or just let them accumulate. And if they're dead, they're like, okay, if every mutation in the spot is dead, it must be important. And then they would look at that. And that's generally how similar thing, like if it's dead, then you look at the SNP or the mutation that caused it. If it's really adaptive, you look at the mutation that caused it. And then you sort of go from there and figure out, okay, what is this mutation doing? Then you learn, what is this protein doing? Then you learn, you do that for every protein. Eventually you figure out what the virus is doing. And that's how, for the most part, how, these images have been made. You go protein by protein and you sort of figure out the whole life cycle. And this is just an example for a mosquito-borne virus that I study. Shell comes in, positive strand RNA, it makes protein, that protein then copies it into minus RNA, that minus RNA makes more positive RNA, that positive RNA also makes the shell proteins, and then the RNA is covered in protein, sent to the cell membrane, and there it gets enveloped and buds off. What I've discussed before, as it adapt to various conditions. Basically, because there's so many viruses, no two blades of grass are the same genome, right? Because every virus is slightly different. The entire population has a bunch of different, what you can call individuals in there. And some of them are better fit for certain conditions. Some of them are less fit for for different conditions. And so they evolve as diverse populations. And the practical consequence of that is that it's allowed the RNA viruses we see today to persist across millions of infection cycles despite evolving host populations. So as a consequence, it can evade. Vaccines are difficult to make because it can evade them quickly. If one single mutation across the entire virus makes the vaccine less effective, it'll just take over the population. Effective drugs must be in sufficient combination. So hepatitis C and HIV drugs, they did not work when you just gave them a single drug because resistant mutations came up. So now it has to be given as a cocktail of multiple different drugs. And it's got a constant threat of species jumping or epidemic potential, as you saw with what happened with HIV, with SARS coronavirus 1. All of these are in the whatever animal they had at that market, or in the chimpanzee in Africa that had HIV. It's not the mutation that allows it to replicate really well in the host animal, but some small percentage of it is just in there. And then when it comes to humans, every so often there happens to be a mutation that makes it much better in humans, and then it catches on in humans. And in the genomics era, we could view this with machines now. That's what I was working on. I'll just skip this stuff. This is what it looks like working on it in the lab. Here's, cells with viruses in it. You basically just take some virus you already have, put it in these cells, and then wait until they die, and then just pour off all the viruses. If it's enveloped, it's now in this red media, which is just, some salt and some stuff to keep the cells happy. And then that's your batch of virus. So now we have the virus to work on. And we do this thing called a plaque assay to see how much viruses grew. You just count these up and multiply by the dilution factor to see how much viruses you have in this thing, if it's enough to get RNA from, if something went wrong, et cetera, et cetera. Then I isolate the RNA, throw it in this machine, and then that gives me all the information for an entire population of viruses. And it just allows us to see viruses evolve in real time. So this is 50 generations of polio. And its frequency here. So basically, if a dot is up here, here's the genome. So this is, it's a 7,000 something base pair genome. Each dot represents a mutation at that spot in the genome and what frequency it is in the entire pool. So 1 is 100%, 10%, 1%, 0.1%, 0.01%, et cetera. And then you could just see the viruses evolve. Some of these dots start doing really well and take over the population because they're adaptive to the conditions I put them under. And this is just seeing natural evolution, natural selection and evolution in action, which is what's going on constantly with viral infections. So there's a variety of different approaches for making the vaccines. And this is what I mentioned before. Like the polio virus, the easiest method is to just passage the virus by weakening it, by just growing it outside of a human for however many generations, and it just accumulates mutations that natural selection does not get out, and then you have the virus just weakened that you re-inject to make antibodies. And inactivated means you weaken it using formaldehyde that just messes up the proteins enough so that it doesn't cause a disease, but your body still recognizes and makes antibodies to it. And both of those strategies are every virus made before 10, 20 years ago is all either this or this. It's really simple to make. I'll show a video on how it's made in a bit. If you guys wanna, whoever wants to watch it, it's not very important, but it's just like a video of how a vaccine is made. It's really simple. The key is the Goldilocks principle. You just gotta like get the right amount of weakening for the body to still recognize and make antibodies, but not cause the actual disease. And several teams are working on this. You have viral vector vaccines, which recombination, meaning you take a piece of DNA from something and insert it into something else, really only became a thing in the late 80s to 90s. So now that's used to make vaccines, where you take, like, I think a common one, a big one right now, like Johnson & Johnson, is where you take the envelope protein, the thing in coronavirus that attaches to cells and therefore you want antibodies against. You take that and put it into a virus, a commensal virus, or a virus that doesn't cause any disease in humans. So like certain adenoviruses are such a mild cold you won't even notice. Or a lot of them are using chimpanzee adenoviruses. So it still like just barely replicates in humans enough to make antibodies, but not enough to cause any sort of disease symptoms. And then you just insert the coronavirus protein in there and hope that, it makes that, and then the antibodies recognize it and do its things. And it's very empirical. It's all like, you just gotta like try it because it's just too complex to predict. So like, this will work in cell culture, but a human or a live organism is just like infinitely more complex. So like, the strategy, you just have to try out different things until you get one. So that's we do this in the lab all the time. And making a vaccine is really easy. None of it is difficult. It's just testing it in humans and figuring out which one's the right one that's difficult. Now you have like more intense ones where like here you still have a virus, right? Now they're just trying DNA. So like instead of the protein shell here and targeting the cells, I don't know why, but they're trying just DNA with the coronavirus gene and just RNA with the coronavirus gene. So they're sort of mimicking like a DNA virus or a plus-stranded RNA virus in that sense, where there is no replication. Now, it's just a piece of DNA or a piece of RNA, and they're hoping to just inject enough that enough pieces of that DNA or RNA gets to where it needs to go to make these proteins. And that's like what's that? Moderna and another company has direct RNA. This is the first time this is being tried. So we have no idea how it'll work. I guess there was a report with some side effects. They're injecting a whole crap load of RNA for this, hoping that some of it, because you can't target it. Here, like the virus itself has all the targeting machinery and it doesn't replicate. So you just have to put everything in there and hope some of it goes in. And then you have like protein-based vaccines where they're just injecting the protein straight up, hoping that enough of it, and doing tricks so that it doesn't get destroyed before antibodies are made to it. Again, there's no targeting, so it's just like the first time they're really trying it, and it's just really difficult. The thing with the direct DNA and direct RNA in this protein thing is that you can make these in an hour. They're so easy to make. So if it works, it'll be great for future as a platform because you can make these immediately, right? You don't have to figure out like what virus it doesn't cause disease in humans, but can still like make the coronavirus gene because not all viruses will make them. So you got to try them out in a bunch of different things. Then you have virus-like particles where it's not like an actual virus. You synthetically add protein and lipids to it. And then you synthetically, there's nothing inside the shell. There's no genome, but you just make the shell with the protein sticking out and hope that the body like recognizes it like a real virus and develops antibodies, which then protect against a real virus. So yeah, I'll drop that in the chat if you want to read up more on that.
I actually just put it on the Slack channel on the random channel in the micro directory, so everyone can have a look. I have one question.
Oh, okay.
I have one question regarding this. So the vaccines. So what is the percentage of success that a company or a researcher will say is acceptable, like it's okay to launch that vaccine into the market. Like it's it works for 80% of the people or 90% or.
I think that's a good question. It changes over time, right? For for smallpox, for example, it just straight up kills a large percent of the population, and like before vaccination, 5% of people who got the smallpox itself died, and that was just like what you did. So as long as vaccinia protected more than, was protective to some degree, and less than 5% of people died, that was good enough. Polio that's one of the reasons children are not given polio vaccines or smallpox vaccines anymore, because they do have side effects. They do have a very small percentage of bad effects. So at the time when people were getting smallpox and getting polio, like that was an acceptable amount of side effects, but now it's not anymore because no one's getting it. So I don't know exactly how they're gonna do this. What they really need is a vaccine to just reduce, at its base level, it just needs to reduce the amount of older people who are dying from the disease by whichever percent. So even if like, I highly doubt one of these new strategies are going to be like the most successful later on. So the first thing just has to reduce it by some amount, and then the next vaccine is now benchmarked against the first vaccine. So then, the next thing that comes out has to be better than the previous one, and so on. So
Okay.
I see it as being an iterative process. I honestly think for the first vaccine, anything above a p-value of 0.05 in the in phase 3 trials is good enough, and a decent enough. I mean, if it has a side effect profile, it wouldn't get past. For vaccines, you don't want a side effect profile, so it wouldn't even get past phase 1. So by the time in phase 3, it's making the antibodies that in cell culture can defeat the virus. It's making it does not have side effects, and it's generally started doing what you want it to do. And then you expand to phase 3, and then you look at efficacy, like on a large scale. And I think as long as the efficacy is above p equals 0.05, it's good enough, because one, it'll just give the population something. Two, that's the minimum you need to like for a company to release it to make their money. And as long as it reduces death rates a little bit, I think that's totally fine.
And there are many diseases that that are still without a vaccine, and they are still killing a lot of people. And some researchers have found like some kind of like treatment in the lab, but when they go to the field and test it, it doesn't work. And an example is like, for example, malaria in South America and tropic countries. It's like a very big issue because it kills a lot of people, especially in the rural areas. And
that's the hardest part, because by the time the first human is injected with these vaccines, it works in cell culture, it works in monkeys, it works in all these other animal models and things that you tested for. But that jump is the biggest, like, we don't know what's happening, we just have to try it scenario. And same thing with, like, there are a bunch of dengue vaccines that were recently tried that for some reason worked all the way through, but once you put it into humans, it doesn't actually reduce dengue. So that failed. And yeah, it is like an art, sort of, as much as it is a science. Kind of like my stick figures.
Is there any
But
other question that you'd like to ask about?
Yeah, I actually, so I wanted to ask, I guess maybe some fallbacks of some of these like methods, because I'm thinking, you talked about, the health of the virus and how how fast it can replicate. Wouldn't that also allow for a lot of like changes? And would it allow for some of these vaccines that are protein-based vaccines? Would the virus be changing the proteins on its envelope that can possibly make these vaccines not work at all?
absolutely, for sure. The question is, and that's true of a lot of different vaccine types. Because when you make these synthetically, there is a mutation rate, so you are making a population of them, similar to like if you put them in an adenovirus or if it's on the virus itself. So like generally, you are giving like some distribution of proteins. The question becomes if it does target the original virus and the virus can escape it with a mutation, what does that mutation do to the virus? So if that mutation like weakens the virus just enough where your immune system could kill the virus, like now your immune system's good to handle it, then that's totally fine. So it has to be a neutral mutation on the virus that allows it to escape the vaccine, for the vaccine to be ineffective. And that you just have to try it, like you could predict certain ones that are really prone to failure in cell culture and in things of that nature, but you really just can't predict what happens in a live human, and so you just have to test it, and it takes years before some of these are discovered. So a bunch of hepatitis B treatments, it takes like a decade before they find out that very slowly that there are treatment escape mutants that have now taken over and the person has hepatitis B again.
Okay. There is one other question about the in the chat. I heard blood type A are more susceptible to severe COVID cases. Why is that? Is that true?
I sort of heard some of it. I don't know how true it is or not. I just haven't looked into it, so I don't know.
Okay.
I could look into it and let you know. But so many people are looking at this that people find all sorts of associations, whether it's correlation or causation. I don't know if we know yet. But if it is causation, I'll look into it, and that sounds interesting. And then Anika, I hope I'm saying this correct, sent me a message saying, a private message saying, are these primarily tested on sick patients or also healthy ones? So that's an interesting question. Now, phase 1 is done in healthy volunteers just to see if there are any side effects. And then, I mean, a vaccine, you can't test it on sick people because it doesn't work that way. You have to give it to them before they get sick. So it's given to a large enough population, and then you look at the statistics between your placebo group and the group given the vaccine to see if, the p-value of 0.05, if this group got the actual disease less than the other group. So what you need, a lot of vaccine trials have failed. There's the reason what you need is a population still getting infected with the disease you're testing for. Like the Ebola vaccines, a bunch of them started and then Ebola just went away. So you don't have enough statistics to do it. So that's true there. But that's only recently. That's like post World War II. Before, the way like the yellow fever vaccine was discovered was different. Now you have like these large clinical trials. But back then, and this happened on Cornell's campus, the Cornell Rockefeller campus, there used to be jails on Roosevelt Island, where Maria lives. And so the scientists would just go to these jails and with their vaccine trials, and then they'll inject like different dilutions or different amount of vaccines into these prisoners. And then, some amount of prisoners will die. And it's like, okay, the vaccine isn't attenuated enough. And then they, like a week or two later, they'll go inject the prisoners with actual yellow fever. And then, some percentage of them will die. And you're like, okay, that vaccine doesn't work. And to some number, they would have gotten the vaccine and not died, and then gotten the yellow fever injection and not died. And then you're like, okay, so it takes like 3 weeks back then to figure out if a vaccine works or not, because you just inject them with the actual thing and see if they die or not. That fell out of favor with good reason after World War II. And so, human testing like that on prisoners or, people like underprivileged groups of that nature does not happen anymore.
So there is still the hospital here in Roosevelt Island. It's a museum, of course, and it's closed, but you can see it from the outside in case you want to do a tourist.
Yeah, and there's really great books. I mean, I got into viruses because I read a book that takes like 2 hours to read. It's a tiny book. I'll give Maria those names, and there's like really good books on the history of all these human experiments and just virus discovery and things of that nature. If any of you are interested, I'll send a chapter, which I'm allowed to send because I'm an editor on this textbook. Don't apologize for having to leave. My bad for my bad for going over. But yeah.
Yeah, feel free to share it to me, and I'll share it to the volunteers, either to Dropbox or Slack.
Yeah, there's a textbook. It's called Principles of Virology, and I guess it's like the commonly used undergrad textbook. Me and a couple other people in the Mason lab, and Charlie's lab, obviously, where co-editors on it, student editors on it. So I can give you the chapter, but please don't spread it around because that'd be, so I'll send it out to you because as a co-editor, I'm not allowed to do that. So I'll give you the intro chapter that goes over like the Baltimore Classification and all these other things and the history of virus discovery and basically what I covered. So a lot of the slides I took from that chapter. So that's a really good resource. Charles has a question. In one of my slides, I showed +1 for archaea. This is from that chapter. So I'll send you guys the chapter. And that question mark, I think, I don't know what it means. I have to, it probably means they've discovered one and there's maybe more that they're not sure of yet because funding for archaeoviruses isn't there. So it takes a while to figure out what archaeovirus they're doing. But good question. I didn't even notice that. And I'm an editor on this chapter. And yeah, so like any other questions?
I have one last question. So if we think that there are viruses wherever there are cells, either bacteria, human cells, any type of cell, have we found viruses in extreme environments, since there are, for example, bacteria in extreme environments, and we know that viruses have been a really key point in the bacteria evolution because they help with the gene transfer and everything. And so my question is, have we found viruses in extreme environments, saline environments, or really hot environments, or any type?
Well, there are viruses everywhere. Like in that one of the books that got me interested in viruses, 1 teaspoon of seawater, when they actually looked at it, had 1 trillion viruses in it. So every teaspoon of seawater has like 1 trillion viruses in there. It's like constantly, the seas have like way more viruses than there are bacteria. And it's like this constant battle that's going on across the entire sea at all times. I mean, your stomach has a ton of bacteria and like pH too. In those stomach, a ton of viruses, ton of bacteria too, but like a lot of those bacteria eating viruses. I don't know of extremely hot environments, but I highly doubt that is any environment where bacteria exist without viruses that eat them. But I'll have to look into it. I'm sure they have some really interesting like physiological adaptations that would be really cool to study.
Definitely.
People don't fund studies of these things typically. Oh, super weird viruses. This guy like just went out on a boat and found these super gigantic viruses. I don't know if you guys have heard of them. They're like the size, you could see them under a light microscope. They only infect amoeba, but he discovered it because he was just on a boat looking for random viruses in the ocean. But extreme environments, I'll have to look into it, Maria. I'll look into it because that's I'm curious now, and that's really cool. I know they exist in low pH for sure because they're all there's a bunch of them in your stomach and stuff. I don't know about extremely hot environments, but I cannot imagine a scenario where there's bacteria and there's not viruses that eat them.
I haven't read anything about that, so I was just wondering if anyone just wasn't anyone curious that just went and looked into it and published something.
Yeah,
But I haven't
I'll
heard.
look into it. Unfortunately, like extreme environment bacteria have are useful. Like PCR, the enzymes are from extreme thermophile bacteria from the Yellowstone hot springs. So they have like biotechnology purpose that is known. Viruses don't. So that field generally just does not get any funding at all. So I'm sure they exist. I don't know how much we know about it yet, just because it's not like a funded thing. And extreme environment bacteria wasn't funded before some guy just went into Yellowstone and picked up that bacteria and revolutionized PCR.
That's the importance of basic science, right?
Yeah. You gotta have some money in there or something.
Yeah.
How do antiviral drugs work? Good question. Each of them is a little different. Let me see if there's a chapter in the book that is exactly, let me look at appendix, antivirals. Let me see if I could pull up, that's not the appendix. Front matter. Yeah. Processing antiviral. There's a really good image here. Maybe I'll send it to Maria and have you, and have her send it to you that describes the different antivirals. But basically, most antivirals work by mimicking a nucleotide. So, DNA and RNA are just polymerase nucleotides. So the individual nucleotides go in ACGT or ACGU. And a lot of antiviral drugs are what are called nucleoside analogs, where they look very close to the nucleotide, but once they're incorporated into DNA or RNA, that RNA cannot be copied further. Those are called like error catastrophes. So like it either cannot be copied further, the DNA or RNA makes a bunch of mistakes, and so like the virus that comes out of it has lower fitness, or they're chain terminators. So once that is incorporated, when the other side is copied, the polymerase just falls off. That's the biggest class of, and the key there is that the viral polymerase incorporates it and dies off, but your DNA does not get too messed up, because that's the balance there. Like, you're making DNA too, you're making RNA too. If it has to be sort of selective, where like the virus polymerase are less complex. So like, they'll incorporate it and die off, but you don't develop cancer by accident. So a lot of them nucleoside analogs. Some of them like prevent the chopping activity, like the virus makes a big protein and then chops it up into individual proteins. Some of them prevent that. There's a large class of them, and they're generally now given in combination. Some of them are prevent, like for HIV, some of them prevent the integration of DNA into your genome. That's a necessary step. Some of them are virus specific, but the largest class are just really tiny small molecules that look close enough to either A, C, G, U, or T. And so the virus puts it into its chain while it's polymerizing. That chain has some defect or another compared to the actual virus. And your polymerase is smarter than that and doesn't really incorporate it, or incorporates it at such a low amount given the duration of treatment that you don't really mess up your DNA copying. And I'll send out a chart that I thought was very good. That's also in this book, highlighting the different drugs out there right now and different different strategies. So the background for this is that the director of the CDC in the 1950s, this guy Telford Work, who discovered a whole bunch of viruses, like hundreds of viruses that were discovered, including I think Zika. He discovered, yeah, and he was the head of the CDC. And obviously, video cameras back then were really expensive, but he liked to make movies, so he just got the CDC to buy an extremely expensive color video camera in the 1950s, and had a plane carried over, you know, it's the government, so whatever, whenever he went to different places to discover viruses. And the entire movie is really interesting. This is from the CDC archives. The previous head of the CDC, I was talking to him, and he was like, "You should watch this movie. It's really cool." And got the DVD from the CDC archives and sent it to me. And the whole movie is about like the process of discovering what virus it is. And this is just the vaccine process, where they called up Walter Reed and it was like, "Hey, we need a vaccine, and this is how it's made. This is how they made vaccines. This is how poliovirus is made. This is how yellow fever is made. This is how all the vaccines before like 20 years ago is more or less is made." So a lot of what you got as a kid is made in this fashion. And let me know, give me a thumbs up if you could hear the audio.
We reported, we set up a questionnaire survey, and this was the evidence that this was a rapidly spreading fatal hemorrhagic fever. So the Rockefeller Foundation consulted with the Walter Reed Army Institute for Research that was already well known for its production of tick-borne encephalitis vaccine for the Russian spring-summer encephalitis. And this is Dr. Edsel, the director, and Dr. Busher, the chief of virology, discussing the request from the Rockefeller Foundation to produce vaccine, mouse brain vaccine, such has been effective in protecting laboratory workers against Russian spring-summer encephalitis. So the protocol includes one lot of mice to be inoculated with the standard Russian spring-summer high passage virus, and another lot of mice with Cassandra forest disease virus that's only 5 or 6 passages from the original isolation from man. And here they're being inoculated with either RSSE or KFD virus. And this is a semi-mass production technique to produce 50,000 doses of vaccine, lead to
Those are the mouse brains.
trituration in the mortar and pestle. Now, this is a standard method of vaccine production with the embryonated egg vaccines, like for influenza and some of the equine encephalitis viruses. The virus is grown in embryos of the chicken egg. Suspensions made in the same way. The Japanese bee encephalitis vaccine used in World War II is made in mouse brain, such as you see being done here with the tick-borne encephalitis and Cassandra forest disease viruses. And over a long period of time, they have learned that a concentration of 0.2% added to the 10% suspension of virus is most effective in inactivating any infectious virus by slow treatment or slow exposure at +4 degrees. So a final concentration of 0.2% formaldehyde is added to the virus suspension and then placed in a +4 refrigeration or cold room
So just a fridge.
for 2 weeks, which they expect will completely inactivate any live virus left. So we have now killed vaccines for Russian spring summer encephalitis and Cassandra forest disease. And these are the actual protocol sheets of the test.
By the way, these are the vaccines that are still
standard
used
Test procedure
for these.
is to inoculate a diluted amount of the vaccine on the first day, the 7th day, and the 14th day in mice. Then they are challenged with the live virus to see if they're protected. But the first lot of vaccine arrived by TWA in Bombay on Christmas Eve. Meanwhile, the Public Health Department of Mysore has brought in the public health officers from all over Mysore to be trained in the administration of this vaccine, which will be given to different populations on a rotating basis.
So that's it. You just inject it into people, and that's your
Here,
vaccine.
The former director of public health, now retired, has been recalled to direct this program. The vaccine is refrigerated in these 1-gallon thermoses, maintaining the cold chain, which maintains the antigenicity of the vaccine.
Refrigeration, very important.
It is now necessary to begin administering the vaccine, and we no longer have responsibility for this public health activity, but we do have responsibility for the research on the effectiveness of the vaccine. Now we're back at Shiga Village, no place better to go than where we first saw the patients that told us they were suffering from the monkey disease. There, the compounder has identified villages where no cases occurred during 1957. And that's where we're recording them by name, having printed books that show the dates on which they received 3 inoculations of the Russian spring-summer vaccine. An initial day, 1 week later, and 5 weeks following the second inoculation. In other words, 3 inoculations of this formalin-killed vaccine. Here's Dr. Amban, who...
Yep. And so that's Russian spring and summer is now called tick-borne encephalitis virus, and this is still the vaccine that's used for that disease. And, you know, that's how you still make influenza vaccines. A lot of different vaccines substitute mouse brains for whatever it grows in. So for influenza, it's generally 9-day-old chickens or chicks in eggs, embryonated chicken eggs, and et cetera, but that's the process. And that's what they're doing for coronaviruses, minus the, you know, rapid human testing part. But that's what's going on with inactivated and attenuated coronavirus vaccines, except they're growing them in African green monkey cells, not mice, or just because it just doesn't grow in mice. They're using cells from monkeys for that. So yeah, hope that was informative. Any questions on there? Let me check the chat. Is there anything on the chat, Maria? I can't. Oh, there you go.
No, just a bunch of thank yous and
Oh,
one.
there were no lab biosafety measures at that time. Do not do this. Do not mouth pipette formaldehyde. You will die if you screw up. There were no gloves while handling. Now you need biosafety level 4 for this virus.
Yeah.
So like there's like 3 facilities in the world where you could do this stuff. But at the time, no gloves, no nothing, mouth pipetting everything. In fact, the virus I studied as a graduate student. What Charlie studied, my PI who won the Nobel Prize, he studied as a graduate student in the 80s. That virus was chosen to study mosquito-borne viruses because when they started, everyone was still mouth pipetting. And if you accidentally sucked in that virus, it didn't kill you. It just caused some cold sores in your mouth. So that's why that virus was chosen as a topic of study. And that's why that virus has like such a long history of study now.
Wow.
So yeah, it's at the time, like if you died, you died. That's that's what it was.
So, okay, if there are no more questions, I'd like to thank you, Pradeep, for your time, for this wonderful talk. It's been like very informative, and I'm sure like many volunteers, all the volunteers that stayed are really passionate about viruses, and feel free to reach out to the micro directory or directly to Dr. Pradeep Ambrose. And thank you so much, everyone, especially you, Pradeep, for your time.
Thank you for inviting me. This was fun. Thanks, Maria. Thanks, Krista. I don't think Krista's on anymore, but thank you so much, Maria. And thanks everyone for attending.
Thank you, everyone. Have a great day. See you, everyone, in the afternoon.